My name is Camila Lopez Ramos. I work at the Channing Division of Network Medicine at Brigham and Women's Hospital and Harvard School of Public Health. So I'm very excited to be here today to talk to you about how we can use gene regulatory network methods to understand sex differences. So to start, why should we care about sex differences? Well, we know there are significant sex differences during development and progression of many diseases, and this will ultimately influence disease incidence and survival. So in this figure, you see a few examples. So there are a lot of autoimmune diseases that have higher incidence in females. As for cancers, we see that most cancer types will have higher incidence in males. Males also have worse prognosis for most cancer types. For infectious disease, we see that some will have a female bias, while other infectious diseases have male bias. So sex differences have been recognized as one of the most significantly understudied aspects of human disease research. Historically, sex has not been accounted for. So if we look in the literature, many papers don't even report the sex of their samples. A lot of experimental studies are done only on males. Even clinical trials have recruited only males in the past. Also, a lot of genomic analysis will exclude sex chromosomes from their analysis, and because there are a lot of challenges to deal with sex chromosomes. So many of the GWAS studies that we have today were done without the sex chromosomes. So in 2015, the NIH introduced a new policy where they stated that sex as a biological variable will be factored into research designs, analysis, and reporting across all studies. And if we're able to really account for sex and also to be able to understand the molecular associated with these sex differences, we can think that we will be able to improve disease prevention and treatment for both sexes. So as I said, there are a lot of challenges when we're studying sex. And I'll focus here more into the genomics research and point out a few of the steps that we need to think about when we're doing a genomic research. So first, we need to ensure adequate sample sizes for well-powered analysis of each sex. We also need to include sex chromosomes in all downstream analysis, even though we know there are some challenges. So one of the challenges is that there are a lot of poor mapping to the sex chromosomes. There are a lot of spurious mapping also in Y chromosomes in female samples. And we're going to talk a little bit more about this on the next slide. And also, we need to think about our statistical analysis. We need to be able to model sex as a biological variable and to perform both sex stratify analysis and sex interaction effects to really understand the effects of sex. So here is an illustration with a few of the steps that we need to look into there. So first is how can we correct this bias that I just talked about that we see in sequencing alignment. So Melissa Wilson's group published a very nice tool to try to correct for this misalignment. It's called the XY alignment tool. And what we need is basically two reference genomes that are informed by sex. So we need a female reference genome and a male reference genome to map our sequencing reads. So in the female, they don't even have the Y chromosome. So we would need to mask the Y chromosome and align the sequences from females in this female-specific genome reference, right? So all the sequences that would misalign to the Y chromosome, they would now be able to align to the correct position in the X. And in males, we need to mask the region that we call PAR, so the pseudo-autosomal region. So this region is exactly the same between the X and the Y chromosome. So it's important to mask them to be able that we have the same dosage, so that in between males and females reference genomes. Next, in letter B, you can see that another important step is to check for biological sex annotation. So here I'm talking about biological sex, an individual with an XX chromosome and another one with an XY chromosome. And we see that in a lot of data sets, there are misannotation of the biological sex. So it's important to look for this and remove this misannotation. And this can be fairly easy done if we do a PCA analysis on the Y gene expression. So if we do this, we expect to see two clusters. So males and females would cluster separately because only males express the Y chromosome genes. And as you can see in this example, there are some samples that are annotated with one sex, but they're clustered with the other sex. So it's important to remove this sample. Finally, in letter C, it's important to think about sex-based analysis. So it's important to think to how we can report our results in a sex-stratified way to sometimes find an effect that is present only in a single sex, like the first plot here. So if we see this is an example of the expression of a genomic marker between normal and tumor. And we see that only in females, showing red, we see an increase in the genomic marker from normal to tumor. So this genomic marker would be efficient only in females, but not in males, showing in blue. We can also find some differential effects between the sex and even some opposite effects. So if you see in the last plot, in females, in red, we see an increase in the genomic marker expression from normal to tumor. But in males, we see a decrease from normal to tumor, right? Imagine we had done this analysis, grouping all samples together and making only one plot. We probably wouldn't even see this effect because on average, the expression in normal and tumor is the same between all samples. And so we know there are many factors, many genetic factors, many environmental factors that can contribute to the phenotypic sex differences that we see. The most obvious one are genetics, showing at the top, so the sex chromosomes are different between males and females. Also the sex hormones, which will impact the expression and a lot of the regulatory process. But there are other mechanisms that are also sex biased. So there are some epigenetic marks, some genes that are differentially expressed between males and females, some sex-biased EQTLs, even some sex bias in the frequency of some SNPs and some mutations found. Also, the way that males and females will respond to environmental exposures can be different, especially in terms of smoking, some chemical and pesticides. So the toxicity and the way that the metabolism responds is different between males and females. So we need to take all this into account. And we just saw that there is this combination of genetic and environmental factors that contribute to sex differences. So our hypothesis is that all these sex and gender bias factors will be reflected in regulatory networks that control gene expression and disease phenotype in each individual. And we can use gene regulatory networks to understand the sex bias gene regulation. And how could we model gene regulation? So let's briefly go over the transcription steps. So we know that transcription factors can bind to each other and form complexes. And we can learn about this type of interaction through protein-protein interaction data. Then the transcription factors will bind to the promoter regions and recruit RNA polymerase II and start gene transcription, right? And we can also learn about these protein-DNA interactions, for example, performing ChIP-seq experiments. Then RNA polymerase will transcribe this gene, and we can measure gene expression through RNA-seq experiments, through microarray experiments. So we could combine these three sets of data, protein-protein interaction, gene expression, and protein-DNA interactions to model a regulatory network. So that's what we're doing. So we have sex-specific gene expression. We combine this expression with other sources of data to model a regulatory network in males and females. And then we can look for differences in network structure. And what will these structure differences in network tell us? You can see at the top here that we'll find some genes to be differentially targeted by transcription factors in males and females. We can even find some pathways to be regulated in different ways between males and females. So this is all important to look for. So I want to show you how we apply this network approach to study sex-specific gene regulation across 29 healthy tissues, and in the second part, how we did this analysis in colon cancer. So to start with the sex differences, we use the GSEX dataset. So we have more than 500 individual and samples across 29 healthy tissues. So in total, we had more than 8,000 gene expression profiles. We combined these gene expression profiles with other sources of data, so the transcription factor binding motifs and the protein-protein interaction data, to model the gene regulatory networks. And we did two main analyses. So we analyzed gene expression differences between males and females, and we also analyzed regulatory network differences between males and females. So to start with the standard differential expression analysis using VIM here, we identified genes that were being differentially expressed between males and females in each of these 29 tissues. You see at the top here is breast tissue. As expected, we have the largest number of differentially expressed genes in breast, followed by adipose, subcutaneous, and thyroid tissues. You'll also see that pretty quick, the number of genes that are differentially expressed will fall down. And if you see on the plot on the right, most of these differentially expressed genes were found in only one or two tissues. The genes that were recurrently found, like across many tissues, there were genes located in the sex chromosomes. So here is the genes that were most differentially expressed across most of the tissues, so the top ranking differentially expressed genes. And as expected, they are sex chromosomes genes. We also look at the pathways, so what were the pathways that these differentially expressed genes were involved with, and we found that a lot of immune-related pathways and a lot of methylation-related pathways were being enriched for differentially expressed genes. But we really wanted to understand the regulatory mechanisms behind this. So how were these genes being transcriptionally regulated? So we use PANDA to generate our regulatory networks. So PANDA will start with a prior network, showing here in the middle, the transcription factor binding site data. So what is this prior network? So this is our best guess of how all transcription factors are connected to genes. And we can look for this scanning the motifs for transcription factor, we can scan the genome for transcription factor motifs located in the promoter regions of genes, right? So we can map all the transcription factor motifs to promoter of genes and then build this prior network. So this will be our initial and best guess of all possible connections between transcription factors and their target genes. And then we can update this prior network with data, right? So we'll integrate expression data and also protein-protein interaction data to really update where these regulatory relationships between transcription factors and target genes are happening. So at the end, we'll have an updated gene regulatory network of transcription factors connected to genes, and an edge weight here will tell us the likelihood that the gene is regulated by the transcription factor. We use PANDA to generate an aggregate network. So this network will represent a population of samples. And then we can use Linus. So Linus uses a bootstrap approach to generate one network for each individual sample. So now we have thousands of female networks, thousands of male networks across 29 tissues. And we can really look at differences between each of these edges. Right? So we compared edge weights between males and females using linear regression models. And we identified a lot of edges to be statistically significant between males and females. So what does this mean? We can think that we have some blue genes, the one shown here. So a blue gene would be a gene with higher targeting of transcription factors in males. We also found the red genes. So these genes would have higher targeting of transcription factors in females. And then we also have the yellow genes. So genes that are targeted by different sets of transcription factors in males and females. So we look for these differentially targeted genes across all tissues. As expected, you can see at the top, the first plot is breast. So breast was the tissue with the highest number of differentially targeted genes, followed by thyroid and brain. At the end here, you see a lot of tissues from the gastrointestinal tract. So these tissues had the least differentially targeted genes. Right? So we found the genes that were being differentially targeted by transcription factors, and then we identified the transcription factors that were driving the sex bias regulation. At the top of this figure, you see the top transcription factors in each tissue that were differentially targeting genes in that tissue. And we also highlight this in this figure by arrows, the sex hormone receptors. And you can notice that the sex hormone receptors are not always at the top of the list. So this tells us that the sex hormones are not the only mediators of sex bias regulation. There are a lot of other transcription factors in play here. So we look next at the pathways that these regulations were impacting. And so we perform a gene ontology enrichment analysis. Here I'm showing the example in brain. So what we found in brain is that a lot of pathways important for brain function, so the regulation of synapse, neurotransmitter transport. So this process, they are enriched for genes that are being targeted by different sets of transcription factors in males and females. When we perform a KEGG pathway enrichment analysis, we found that diseases such as Parkinson's and Alzheimer's are also being targeted by different sets of transcription factors in males and females. And these are diseases that are known to have sex bias manifestations. I want to show another example in heart. So in heart, we also found the heart process and cardiac cell development. These are processes important for heart function that are being regulated by different transcription factors in males and females. And also cardiomyopathy is being regulated by different transcription factors. The last example that I wanted to show today is thyroid. So a lot of the immune process in thyroid are being differentially targeted between males and females, and also the autoimmune thyroid disease, another disease with sex bias manifestation, is being differentially targeted between males and females. So I want to give an important note here. If you remember, these are all healthy tissues, right? So what does it mean to have this sex bias associated with some disease pathways? What it means is that even at baseline, so if we look in healthy tissues, genes that are important for diseases, they are being regulated in different ways in males and females. And this might become really important to understand sex bias in disease development. So maybe after a specific exposure or a mutation, we would expect that the networks of males and females will respond different to that impact. And this will impact disease incidence and how disease will progress in each sex. Right? So to conclude, we saw that sex differences are evident in tissue-specific gene regulatory networks. We also found that even though most transcription factors were not differentially expressed between males and females, they were targeting different genes. Right? And these genes that were being differentially targeted between the sex, they were enriched for disease and tissue-related functions. Finally, I just want to mention, if you're interested, if you want to download these networks and do further analysis, they are all available for download at Grand Network Medicine. So now let's talk about how we use the similar network approach to study sex differences in colon cancer. As I mentioned, males, for most cancer types, they will have higher risk and higher mortality rates than females. Also true for colon, which is the focus now. In colon cancer, we also see that females have higher survival benefit after chemotherapy treatment. And as we saw before, there are a lot of factors that can impact sex differences. So we can see environmental factors, sex hormones, and all the combination of genetics, epigenetics, and gene regulation. This will ultimately impact cancer incidence and even response to treatment in males and females. So in this work, we used data from the TCGA dataset as our discovery set, and we also used 5 independent studies that were available in GEO as our validation set. So we have a robust pipeline to process this RNA-seq data and microarray data and also perform some gene expression normalization. But I want to again emphasize that one important step is to remove sex misannotated samples. So we performed a PCA on the expression of the Y gene genes, and we see that some samples annotated in one sex, they are clustered with the other sex. So we removed these samples that were misannotated for their biological sex. So at the end here, we have 445 colon cancer samples. These are all primary tumor samples before any treatment. And in our validation set, we have more than 1,000 colon cancer samples. We started with a differential expression analysis. So again, here we used VUM. And what we found is that the genes that were differentially expressed between males and females are located on the sex chromosomes. So not a lot of new biology from this standard analysis. But we wanted to really understand the regulatory mechanisms. And we performed a similar analysis. So we used PANDA, and then we used Linus, and we generated gene regulatory networks for each individual sample. And then we compared the male and the female networks. So how can we compare them? So one way is to calculate their gene targeting score. Right? So what is the gene targeting score? It's the sum of all the edge weights around this gene. It's the gene in degree of the network. And what they tell us, the gene targeting score will tell how strong this gene is being targeted and regulated by the transcription factors. We can then compare the gene targeting scores between males and females using linear regression model and correcting for covariates to find the genes that were being differentially targeted between males and females in colon cancer, and finally the pathways they were enriched for. And for this, we perform a gene set enrichment analysis using GSEA. So the first cool results here is that we find sex differences are also present in autosomes. So a lot of autosomes are being regulated by different sets of transcription factors. And when we look at the pathways, we also find a lot of interesting pathways. So pathways important for cancer, such as NOX signaling, Wnt, mTOR. So these pathways were enriched for genes with higher targeting in males. When we look at the drug metabolism pathway, we saw that these pathways were enriched for genes with higher targeting in females. So here's a representation of the drug metabolism genes showing as circles. And you can see that all these drug metabolism genes have higher targeting by females, shown by the purple edges. I also want to point to an interesting case here, the CYP gene. So if you see a CYP gene is targeted by both green and purple edges. So what this means is that different sets of transcription factors are regulating CYP in males and females. If you can read this, a lot of the purple edges are estrogen receptors. So this would be transcription factors active to regulate CYP only in females. We repeated the analysis in our validation set, and again, we confirmed that genes in the drug metabolism pathway were more strongly targeted in females. When we look at these genes, we know that there are a lot of them that are highly polymorphic genes. A lot of them have been already associated with colon cancer risk, and a lot of them belong to the glutathione S transferase family. So these GST families are families that are important to neutralize and eliminate toxic compounds and xenobiotics. Okay? So we found out these differences in the gene regulatory networks around the drug metabolism genes. And this was in primary tumors even before any treatment. So we could expect that these differences would ultimately impact the survival outcome in males and females, right? So that's what we did next. So we performed a survival analysis in males and females that were treated with chemotherapy. In this analysis, we stratified the patients based on how strong their drug metabolism pathway was targeted. And what you can see here in purple is that in females with high targeting of the drug metabolism pathway, they were associated with a significant higher overall survival. All right? So to summarize, what this means is that females with a more male regulatory pattern will have a worse survival, right? And this is in a primary tumor, even before any treatment was done. So this will tell us that males and females are already primed to respond differently to treatment, and this will ultimately impact their overall survival. So to conclude, colon cancer has significant sex differences related to transcription factor regulatory patterns. We found that genes involved in drug metabolism pathway were more strongly targeted in females, and this indicated that higher targeting of the drug metabolism pathway was associated with a better overall survival in females treated with chemotherapy, but not in males. So I just want to finish with some resources if you are interested in doing some network analysis. We have all the code available in our NetZoo platform. So here you find tools to reproduce the Panda networks and the Linus networks. We also have a lot more tools to analyze networks available here in NetZoo. If you're interested in working with the network models that were already generated, you can download them all in Grant. Not only the networks that I showed here, but in Grant, there are also a lot more cancer networks available. There are networks for cell lines, and even cell lines that were treated with drugs. And finally, if you want to do some network analysis, but you don't know where to start, we have a lot of tutorials available in NetBooks. And these tutorials will help you to use the NetZoo tools, will help you to recapitulate a lot of the analysis and plots that we have published before. And you can even, if you're interested, use your own data and generate networks on the cloud. So you can use NetBooks to also analyze your own data with the NetZoo tools. So just to finish with some take-home message, we found that gene regulatory differences between the sex occur in every tissue. So we found this in healthy tissues and also in cancer. So this tells us that sex bias regulation of cellular process is systemic. Understanding the molecular mechanism that drives sex differences in cancer is essential if we are to develop effective and sex-aware precision cancer care. Also, network methods can provide an effective framework to investigate sex differences in gene regulation and can identify distinct regulatory processes that are active in males and females. So these network methods can easily be expanded to other complex diseases. So to finish, I'd like to acknowledge all the people involved in this work, especially Dr. John Quackenbush and Dr. Don DeMeo. And thank you for your attention. I'm happy to take any questions.